Hi dear students, welcome back to Excel Rapid Focus series. I am your Nipun. Today we are going to discuss the chapter Biotechnology Principles and Processes, a big chapter. Okay, so let's do the questions. Listen. As usual, we are going to discuss the most important and most repeated questions of this chapter. Okay, if you listen carefully, you will get full marks in this chapter. Okay, so let's do the first question. Write the name of the enzyme that are used for isolation of DNA from bacterial and fungal cells respectively for recombinant DNA technology. You know that in recombinant DNA technology, the first step is isolation of DNA. The first step is isolation of DNA. In recombinant DNA technology, the first step is isolation of DNA. Right? What is isolation of DNA? This is a cell, this is a cell. Okay, if that a bacterial cell, fungal cell or plant cell, that cell will have a cell wall. Okay, cell wall, cell membrane. Inside that there will be cytoplasm. This is nucleus. Inside the nucleus, here is DNA. Okay, taking out DNA is called isolation of DNA. This is the first step of recombinant DNA technology. Okay, for isolation of DNA, we have to do many things. Okay, we have to do many things to isolate DNA. See, the first step of isolation of DNA is breaking the cell wall. Breaking the cell wall. Okay, first we have to break the cell wall. Okay, second step, we have to remove the impurities. Removing impurities. Okay, third step, we have to precipitate the DNA. We have to precipitate the DNA. Okay, and the fourth step is spooling. So which are the steps of isolation of DNA? Breaking the cell wall. Second one, removing the impurities. Third one, precipitation of DNA. And last step is spooling. Got it? Listen, for that, take the cells in a test tube. Okay, take the cells in a test tube from which we want to isolate the DNA. So to isolate DNA, as a first step, we have to break the cell wall. Okay, to break the cell wall, we have to add the cell wall digesting enzymes to the test tube. Okay, see, if we are taking the DNA of bacterial or plant cell, see, plant cell, you know the cell wall of plant cell is made up of cellulose. In order to break cellulose, we have to add cellulase. To break the cell wall of plant cell. But if we are taking fungal cell, okay, the cell wall of fungus is made up of chitin. So what should we add to break the cell wall of fungus? Chitinase to break the chitin. Okay, but if we are taking a bacterial cell, see, the bacterial cell wall is made up of polysaccharide and amino acid. Okay, or we can say NAG and NAM. Okay, in order to break the bacterial cell wall, we have to add lysozyme. Okay, now listen, write the name of the enzymes that are used for the isolation of DNA from bacterial and fungal cells. From bacterial cells, we can use the lysozyme, and for breaking the cell wall of fungal cell, we can use chitinase. Okay, so you got the answers. Got it? Okay, let's move to the next question. Explain palindromic nucleotide sequence with the help of suitable example. Explain palindromic nucleotide sequence with the help of suitable example. See, palindrome, you may have heard about palindrome. Okay, palindrome letters are there, palindrome numbers are there. Okay, for example, Malayalam is a palindrome letter. If you read from here to here, you can can see, you can read Malayalam, and from here to here also it reads the same Malayalam. And palindrome numbers are there 1 2 3 4 3 2 1. If you read from here to here and here to here, the same. Okay, such things are called palindrome. But palindrome sequence are in case of nucleotides. Okay, palindrome sequences are the nucleotide sequences. Palindrome sequence are the nucleotide sequences which read the same from both ends on opposite strands. Okay, this is the definition of palindrome sequences. Palindrome sequences are the nucleotide sequences which read the same from both ends on opposite strands. For example, listen, this is a nucleotide chain. Okay, the nitrogen bases are GAATTC, guanine, adenine, thymine, cytosine. Okay, and on the opposite strand, you know that the complementary nitrogen base of guanine is cytosine, complementary of adenine is thymine, on the opposite of thymine it is adenine, here also guanine. Got it? So if you read, see, this is the 5' end of this nucleotide, this is the 3' end of this nucleotide, and here it is the 5' end, this is the 3' end. Okay, on the upper strand, if you read from 5' to 3', you will get GAATTC. And if you read the below below one, okay, this one from 5' to 3', you will get the same GAATTC. Got it? So GATTC, it reads the same from both ends on the opposite strands. This is called palindromic sequence. Now you got the example for palindromic sequence. Got it? And what is the peculiarity of palindromic sequence? Palindromic sequences are recognition sequences for restriction enzymes. Restriction enzyme will come and cuts in the palindromic sequences. Okay, GATTC is the palindromic sequence. At the same time, it is the recognition sequence for the enzyme, the restriction enzyme, echo R1. See, echo R1 is a restriction enzyme. Okay, it cuts somewhere in the recognition sequence, somewhere in the palindromic sequence. You know the location of cutting of echo R1. Echo R1 cuts between G and A. Echo R1 cuts between G and A. Okay, echo R1 cuts between G and A. Got it? And after cutting this, the portion will be single-stranded overhanging stretches. Got it? After cutting, the portions will be like this. Okay, one piece and the other piece will be like this. Okay, here G, C, T, T, A. Here also A, T, T, C, G. Okay, got it. Now these ends are single-stranded overhanging stretches. Okay, they can be combined by using ligase. So these are sticky ends. Got it? Okay, so the question is explain palindromic nucleotide sequence with the help of suitable example. The suitable example is GATTC. It is a recognition sequence of the restriction enzyme echo R1. Okay, one more question I am adding with this. Which is the first restriction enzyme? That is HindII. Which is the first restriction enzyme? That is HindII. Okay, and one more thing I am adding, you must know the criteria of naming of echo R1. That's a very simple question. You know that the naming of echo R1. Okay, the first letter is from the first letter of genus name, okay, of the bacteria. Second and third letters from the first and second letters of species name of the bacteria. R, this fourth letter indicates the strain of the bacteria, and this Roman number indicates order in which the enzyme is isolated. You know that. Okay, so we are going to do the next question. Name and explain the technique used for separating DNA fragments and making them available for biotechnology experiments. Come on, name and explain the technique used for separating DNA fragments and making them available for biotechnology experiments. Separation of DNA fragments by a technique called gel electrophoresis. Gel electrophoresis. Gel electrophoresis. Gel electrophoresis. Okay, gel electrophoresis. You know that. What is gel electrophoresis? Separation of DNA fragments based on their size in an electric field. Okay, separation of DNA fragments based on their size in an electric field. Okay, so we have to take agarose gel in the device of gel electrophoresis. This is the device of gel electrophoresis. Okay, we are adding agarose gel. We are adding agarose gel. We are adding agarose gel. Agarose gel is taken from seaweeds, gelidium and gracilaria. Okay, so now to the agarose gel we are dipping a comb, and there forms some pores called wells. Okay, these are wells. Okay, now we are connecting the ends of the device on a power supply. Okay, positive charge on negative charge on one end and positive charge on the other end. Okay, to the well side we are connected the negative terminal of battery, and to the opposite side we are connected to the positive charge of battery. Now we are adding DNA into the wells. We are adding DNA fragments into the wells. You know that DNA fragments are negatively charged. DNA fragments are negatively charged. Okay, now these negatively charged DNA fragments have a tendency to move towards the positive charged part, or anode. Now these negatively charged DNA fragments have a tendency to move towards the anode. Okay, these DNA fragments will move through the sieving effect of the gel. Okay, and smaller DNA fragments can move farther through the small sieves of the gel. Smaller DNA fragments will reach here faster. Okay, and comparatively larger one will be here. Okay, smaller fragments will be here and larger fragments will be here. Now the DNA fragments are separated based on their size in an electric field. Okay, this is gel electrophoresis, but the problem is we cannot see the DNA fragments. DNA fragments are that much smaller, and also in this agarose gel we cannot see where is our DNA fragments gone. Okay, so in order to visualize this DNA fragments, we have to add ethidium bromide. We have to add ethidium bromide. We have to add ethidium bromide into that agarose gel. After adding ethidium bromide, we have to give UV light. We have to give UV exposure. Once again, in order to visualize the DNA fragments in agarose gel, we have to add ethidium bromide into the gel. After that, we have to give the UV exposure. Then you can see the DNA fragments in bright orange colored bands. We can see DNA fragments in a bright orange colored bands. Okay, now DNA fragments are visualized. Now you can take out the DNA fragments from gel piece. Okay, now you can take out your desired DNA fragment from gel piece by a process called elution. See, for biotech, name the experiment, explain and technology used for separating DNA fragments and making them available for biotechnology experiments. The process is gel electrophoresis, and extraction of DNA fragment from gel piece is by elution. Got it? Okay. Now listen, in a pathological lab, a series of steps were undertaken for finding the gene of interest. Describe the steps or make a flow chart showing the steps of amplification of gene of interest. Okay, amplification of gene of interest. Amplification of gene of interest. Amplification of gene of interest. Okay, what is mean by amplification? Amplification means making multiple copies. Amplification means making multiple copies. See, if you have a piece of DNA, we have to make multiple copies of that. We have to make multiple copies of that. This making of multiple copies of DNA is called amplification. Got it? And this amplification is done by a process called PCR. This amplification is done by a process called PCR, polymerase chain reaction. Polymerase chain reaction. Got it? Now look at the question. Describe the steps or making the flow chart showing different steps of amplification of gene of interest. There are 3 steps involved in amplification. Which are the 3 steps involved in amplification or PCR? First step is denaturation. First step is denaturation. Okay. Second step is annealing. Okay, third step is extension. Which are the 3 steps of PCR? Denaturation, annealing, extension. Okay, now I am going to explain the steps. Listen. First, take the DNA fragments that you want to get amplified. Okay. This is the desired DNA fragment. This is the polarity 5 prime, 3 prime, 3 prime, 5 prime. Okay, if you give high temperature, the 2 strands will be separated. That process is called denaturation. Denaturation is the separation of 2 strands by giving heat. Okay, if you give high temperature like almost 94 degrees Celsius, the 2 strands will be separated. That step is called denaturation. Got it? Okay, now as a next step, we have to add primers to that. Now, primers will be added. Listen. First of that, listen. This is 5 prime end. This is 3 prime end. This is 3 prime end. This is 5 prime end. Okay. Now, primers will be added to the 3 prime end of the strands. Okay, now primers will be added to the 3 prime ends of the strand. Okay, these are primers. This is primer. Okay, this is primer. Got it? Okay, this is the 5 prime end of the primer. This is the 3 prime end of the primer. Okay, this is the 5 prime end of the primer. This is the 3 prime end of the primer. Okay, now listen, this primer adding process is called annealing. So, primer adding process is called annealing. Got it? Next step, listen. This is one template strand. This is the other strand. 5 prime end. 3 prime end, 3 prime end, 5 prime end. Okay, now the primers. 5 prime, 3 prime, here also 5 prime 3 prime. Got it. Now see, listen. The enzyme involved in polymerase chain reaction is DNA polymerase enzyme. Okay, DNA polymerase enzyme. DNA polymerase catalyzes the DNA polymerization. Okay, listen. See, this DNA polymerase enzyme bind with the primer. Okay, and adds new nucleotides to the primer. Okay, the nuclease, sorry, DNA polymerase enzyme adds new nucleotides to the primer. And extends the primer. That is called extension. Once again, listen. The DNA polymerase enzyme comes and binds with the primer, and that DNA polymerase enzyme adds new nucleotides on the primer and extends the primer. This is called extension. Now you got two DNA fragments. We have started with one desired DNA fragment. Now you got 2 copies of that. Okay, now if you continue the process again, the 2 will be 4 will be 8 will be 16, like that. And if you do the 30 cycles of this PCR, you will get approximately 1 billion copies. Got it? So once again, the three steps of PCR is denaturation, annealing, extension. Okay, and the enzyme used is DNA polymerase enzyme. The DNA polymerase enzyme, see, formerly the DNA polymerase enzyme were not heat resistant. Okay, later the scientists invented a thermostable DNA polymerase enzyme. That is called Taq polymerase. The full form of Taq is Thermus aquaticus. Okay, see, the thermostable DNA polymerase enzyme Taq polymerase is extracted from a bacteria, Thermus aquaticus. Okay, this Taq polymerase is heat resistant. Okay, which means the Taq polymerase can tolerate high denaturation temperature. Got it? Okay, so let's move to the next one. Name the source organism that possesses Taq polymerase. What is special about function of this enzyme? See, Taq polymerase is a thermostable. It is resistant to high temperature. Okay, and this Taq polymerase is extracted from which one, guys? From which bacteria? Thermus aquaticus. Okay. Next question. Name any 2 natural cloning vectors. Give reasons that make them act as cloning vectors. Write the 2 characteristics that engineered vectors are made to possess. Okay, now listen. It is very easy. Name 2 natural cloning vectors. Come on. Which are the natural cloning vectors? First one is plasmid. Plasmid is a natural cloning vector. Okay. Second one is bacteriophage. These are the 2 natural cloning vectors. Okay, and give reasons that make them act as cloning vectors. For every cloning vector, there should be 2 advantages or 2 characters. First one, ability to enter into a host. Okay. Second one, ability to replicate within the host. Okay, see, this is a host cell. Okay, host bacteria. Okay, this is the chromosomal DNA of bacteria. Listen, see, this is a vector. This vector must have the ability to enter into the host cell. Ability to enter into the host cell. Second one, ability to replicate itself in the host cell. Replication means making copies. Okay, ability to replicate within the host cell. These are the advantages of a cloning vector. Got it? So write the two characteristics the engineered vectors are made to possess. Can you name any artificial cloning vector or any engineered cloning vector? Yeah, engineered cloning vector is PBR 322. PBR 322 is made by Bolivar and Rodriguez, the engineered cloning vector. Okay, the engineered cloning vectors has two specialities. First one. See, it is easy to link. It is easy to link a foreign gene. It is easy to link a foreign gene. It is easy to link a foreign gene. Okay, second advantage, easy to select recombinants. Easy to select recombinants. Okay, listen, write the two characteristics the engineered vectors must possess. These are the two characteristics of an engineered vector. First one, it is easy to link a foreign gene. Second one, it is easy to select recombinants. Got it? Okay, so let's go to the next question. Name the organism in which the vector shown is inserted to get copies of the desired gene. Name the organism in which the vector shown is inserted. See, the given vector is PBR 322. Okay, it is a genetically engineered plasmid vector. Usually, plasmid vectors are introduced into bacteria. So the host organism is bacteria. The answer of first one is bacteria. Name the organism in which the vector shown is inserted to get the copies of desired gene. The host organism is bacteria. Got it? Second one, mention the area labeled in the vector responsible for controlling the copy number of the inserted gene. Okay, come on. Which part, which DNA sequence is responsible for controlling the copy number? Which one? You know that ORI site controls the copy number. There are three features for a cloning vector. First one, ORI site. Okay, or origin of replication. ORI site or origin of replication. Okay, this ORI site is the site from where the replication starts, and ORI site controls the copy number. Okay, second one, cloning sites. Cloning sites. Okay, cloning sites are these. Listen. PST1, PVO1, EchoR1, CLA1, HIND3, BAMH1, SAL1. These are cloning sites. Cloning sites are the restriction sites for restriction enzymes. Okay, see, the restriction enzymes will come and cuts in their specific cloning sites. For example, if we use EchoR1 as the restriction enzyme will cut in this region. Okay, if we use BAMH1 as the restriction enzyme, that restriction enzyme comes and cuts in this site. Got it? So restriction site or cloning sites are the sites where restriction enzyme cuts and foreign DNA binds. Got it? Third one, selectable markers or marker genes. Okay, see, AMP-R, here you can see AMP-R, ampicillin resistance gene. Here you can see TET-R or tetracycline resistance gene. Okay, these are the two antibiotic resistance gene. They help to select recombinants from non-recombinants. So they are called selectable markers. Once again, AMBR and TET-R are the two antibiotic resistance gene. Okay, they help to select recombinants from the non-recombinants. That's why they are called selectable markers. Got it. Okay, so these are the three features of a cloning vector, and the copy number is controlled by ORI site. So the answer for second question is ORI site. Third one, name and explain the role of selectable marker in the vector shown. What is the role of selectable marker? They help to select recombinants from non-recombinants. Got it. So next question. Describe the characteristics that a cloning vector must possess. You know which are the three characteristics of a cloning vector. First one, ORI site. Second one, cloning sites. Third one, selectable marker. We have seen that already in this question. Okay, these are the features of a cloning vector. Second one, why DNA cannot pass through the cell membrane? Explain how is a bacterial cell made competent to take up recombinant DNA from the medium. Okay, listen. This is a bacterial cell. This is a bacterial cell. Okay, this is a DNA. Okay, you know that the cell membrane is hydrophobic. Cell membrane is hydrophobic. Why? Cell membrane is made up of lipids and proteins. Okay, the lipids are hydrophobic. See, the cell membrane is hydrophobic, but DNA, this is DNA. Okay, DNA is hydrophilic. Okay, this hydrophilic DNA cannot pass through the hydrophobic membrane, or the hydrophobic membrane will not allow any hydrophilic substance to pass through. Got it? Yeah, that's the answer. And why DNA cannot pass through the membrane? Because DNA is hydrophilic and the membrane is hydrophobic. Next one, explain how is a bacterial cell made competent to take up a recombinant DNA from the medium. See, if we want to introduce a recombinant DNA into a host, we have to make the host cell competent. How to make the host cell competent? You know, there are 4 steps to make the host cell competent. Okay, I'll write here the 4 steps. First one, treating with the calcium ions. Treating the host cell with calcium ions. Second one, incubating the bacterial cell on ice. Third one, giving heat shock to the cell. Heat shock. Okay, giving heat shock at 42 degrees Celsius. Okay, just transferring the bacterial cell from ice to heat, that is called heat shock. 42 degrees Celsius temperature. Again transferring the bacterial cell into ice, incubating on ice again. These are the 4 steps to make the host cell competent to take up the DNA. Got it? Okay, so let's move to the next question. In PBR 322, this is PBR 322, foreign DNA has to be introduced in TET-R region. This is TET-R region. Okay. From the restriction enzymes given below, which are those PVU1, EcoR1, BAMH1, which one should be used and why? Okay, if we want to introduce a foreign gene in TET-R antibiotic resistant gene, which restriction enzyme we should use? The answer is BAMH1, because in TETR only BAM H1 restriction site is present among the three. Okay, and the restriction site for PVU1 is in amp R. Restriction site for echo R1 is in this region. Okay, so the restriction site for BAM H1 is present in TETR region, so we can use BAM H1. Got it? Okay, next one, give reasons why the other enzymes cannot be used, because the other enzymes, the PVU1 and echo R1 cannot be used because the sites for those enzymes are not in the TETR region. Okay, yeah. Next question, explain how recombinants and non-recombinants are differentiated on the basis of color production in the presence of a chromogenic substrate. Name that procedure. Before that, listen to this question. See, why does the insertional inactivation method to detect recombinant DNA is preferred to antibiotic resistance procedure? Okay, see, we have to do this question to do the previous question. Okay, listen, what is insertional inactivation? I'll tell you what is insertional inactivation. Listen here. You know, this is PBr 322. This is PBr 322. Okay, I'll write the definition of insertional inactivation here. Insertional inactivation. Insertional inactivation. Okay, listen. Inactivation of a gene. Inactivation of a gene due to the insertion of a foreign gene. Due to the insertion of a foreign gene. What is insertion inactivation? Inactivation of a gene due to the insertion of a foreign gene. Listen, here TETR, tetracycline resistant gene is here. The tetracycline resistant gene gives resistance against the antibiotic tetracycline. Okay, tetracycline resistant genes give resistance against antibiotic tetracycline. Okay, and ampicillin resistant genes give resistance against antibiotic ampicillin. Got it? Okay, now listen, if I use BAM H1 as a restriction enzyme, the BAM H1 will come and cut at this site. Okay, now the TETR gene is broken. Got it? And to this site, I am adding a foreign gene. To this site, I am adding a foreign gene. See, a foreign gene is inserted. A foreign gene is inserted. Now what happened? The TETR gene is inactivated. Okay, so the inactivation of TETR due to the insertion of a foreign gene. Okay, listen, what is insertion inactivation? Inactivation of a gene due to the insertion of a foreign gene. Got it? And what if I add a gene in the PVU1 site? What if I add a foreign gene in the PVU1 site? The AMPR gene will be inactivated. Got it? Inactivation of AMPR due to the insertion of a foreign gene. Inactivation of a gene due to the insertion of a foreign gene is called insertional inactivation. Got it? Okay. Now listen. Okay. Why does the insertional inactivation method to detect recombinant DNA is preferred to antibiotic resistance procedure? Okay, see, we can select the recombinants by two methods. One by using antibiotic resistance, and second one is by using insertional inactivation. Basically, the two methods are based on insertional inactivation. Okay, so first in your book, first we have to study the selection by using antibiotic resistance method. Let's see how the recombinants are selected by using antibiotic resistance method. Listen. See, this is a culture medium of bacteria. This is a culture medium of bacteria. It contains many bacteria. Okay, even though I am drawing only three types of bacteria. Okay, see, now I am taking the PBr 322. Okay, and I am using BAM H1. I am using BAM H1 restriction enzyme. You know, the BAM H1 restriction enzyme will cut in the TET-R. In the TET-R. Listen. BAM H1 restriction enzyme cuts in the TET-R region and inserting foreign gene into the TET-R region. Now what happens? The TET-R, tetracycline resistant gene, will be inactivated due to the insertion of a foreign gene. Got it? Okay. And some vectors may not be cut by this one. Got it? Now we got recombinant DNAs and non-recombinant DNAs. Okay, many recombinant DNAs we have and many non-recombinant DNAs we have, and we are adding the non-recombinant and recombinant DNAs into the medium of bacteria. Okay, now some bacteria will take the non-recombinant DNA. Okay, some bacteria will take recombinant DNA. Some bacteria will take recombinant DNA. Some bacteria will take recombinant DNA. Okay. Okay. These two bacteria taken some DNA. Got it? They are called transformant. They are called transformant. They are called transformants because transformation occurred to them. Okay. See, these are transformants. These are transformants. Got it? And this bacteria didn't take any DNA, so it is non-transformant. 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 Got it? And among the two transformant, one is recombinant transformant. Recombinant transformant. Recombinant transformant. Got it? And this one is non-recombinant transformant. Non-recombinant transformant. Non-recombinant transformant. Non-recombinant transformant. Got it? So in this medium we have got three types of bacteria, not three bacteria, three types of bacteria. One is recombinant transformant, second one is non-recombinant transformant, third one is non-transformant. Okay, see, in this, among the three we have to select the recombinant transformants. Okay, the recombinant transformants. How can we select the recombinant transformants among the others? How to eliminate the others and how to select the recombinant transformant? Yes, first we have to add ampicillin to this medium. First we have to add ampicillin. Ampicillin to this medium. Then what happens? When ampicillin is added, the bacteria which does not have the ampicillin resistant gene will die. Who will die? This non-transformant will die because the two remaining bacteria have the plasmid and it possesses ampicillin resistant gene. It does not have the plasmid and it does not have ampicillin resistant gene. So when ampicillin antibiotic comes, this one will die. Got it? And the one will be selected. Now we have to select recombinant transformants by eliminating non-recombinant transformant. How to do that? See, listen. See, this is the culture plate. Okay, this is the culture plate. Already this bacteria is eliminated. Now you have two colonies of bacteria. Okay, and one thing is sure, among them one is recombinant transformant and the other one is non-recombinant transformant. Okay, this is your original culture medium. Now you have to make a copy of this. Okay, copy of this. Okay, we have studied how to make the copy, how to make the replica plate in your class. Replica plate. Okay, this is replica plate. Okay, replica plating, you don't have to study the method of replica plating. Okay, anyway, we are taking the copy of this medium, replica plate. Got it. Now to this replica plate we are adding tetracycline is adding. Okay, when we add tetracycline, see, if we are adding tetracycline to this, who will die? Who will die? Who do you think die? Come on, you know that the transformants will die. The transformants will die. Why? Because we added foreign gene into the tetracycline resistance gene. Okay, if foreign gene is inserted, the tetracycline resistance gene will be inactive. Okay, so the recombinants will not have resistance against tetracycline. So if we are tetracycline, the recombinants will die. Okay, we can see one colony is dying in the replica plate. Okay, by locating the position of the eliminated ones in the original plate, we can select them. Okay, in the replica plate, one colony become dead, that is recombinant, and from the original plate, by locating the position, we can easily select the recombinants. Now the recombinants are selected by a method called antibiotic resistance method, but this method is a cumbersome process. It is a difficult process. Why it is considered as a difficult process? Because it requires culturing of bacteria alternately in a two different medium. Okay, first culturing the bacteria in ampicillin containing medium, and the non-transformants will be eliminated. After them, culturing in another medium containing tetracycline. Then who will be selected? The recombinant transformant will be selected. Since it takes two steps, it is very difficult process. Okay, but we want to select the recombinant transformants in a single step. For that, we have another method called antibiotic resistance method. Okay, I'll show you what is antibiotic resistance method. Listen. Okay, guys, listen. This is a culture medium. In this, we have three types of bacteria. Okay, one is non-transformant, one is non-recombinant transformant, one is recombinant transformant. Okay, see, this one does not have any plasmid. Got it? These two have the plasmid, so these are transformants. This one is non-transformant, and among these two, this one is recombinant transformant. Got it? But in this method, in insertional inactivation method, selection by insertional inactivation method, we are using another cloning vector, not the PBRC22. Instead of PBRC22, we are using another cloning vector. The name of that is PUC19, plasmid of University of California 19. Okay, see, this is the PUC19 cloning vector. Okay, now here there is ampicillin resistance gene. Okay, and here there is a gene called LAC-Z. Okay, LAC-Z is a gene that codes for beta galactosidase enzyme. LAC-Z is a gene that codes for beta galactosidase enzyme. LAC-Z is a gene that codes for beta galactosidase enzyme. Okay, now listen. Taking a copy of this. Okay, taking a copy of this. Now we are cutting the lac-Z gene and introducing foreign gene into the lac-Z gene. Okay, now foreign gene is inserted. Now what happens? If you insert a foreign gene, the lac-Z gene will be inactivated. Okay, which means now this vector is not able to produce beta-galactosidase enzyme. Got it, got it. Listen, this beta-galactosidase enzyme, when combined with a chromogenic substance, makes a blue color. See, the beta-gal when combining with a the beta-galactosidase enzyme when combining with a chromogenic substance produces a blue color. Got it, got it. Okay, now you got the basic thing, and now we have to do the process. Okay, so once again we have the culture medium. We have three types of bacteria. One is non-transformant, another one is non-recombinant transformant, third one is recombinant transformant. Okay, the plasmid we used is PUC90. It has ampicillin resistant gene and lac-Z gene. Got it. Now to this we are adding ampicillin. If you add ampicillin to this, who will die? A, B or C? Who will die? If you add ampicillin, C will die because it does not have ampicillin resistant gene. Otherwise it does not have what? The plasmid. Okay, so the third one will die because of the absence of ampicillin resistant gene. Okay, these two will survive. Okay, and to the same medium we are adding a chromogenic substance. Okay, after some time we can see one colony turns into blue color. If blue color is present, we can identify it is recombinant or non-recombinant. See, I'll tell you the method. Come on. This is recombinant. Okay. Sorry, we not yet. We still now we don't know which one is recombinant and non-recombinant. Okay. See, there is an insert here. Got it. If there is an insert, which means inactivation of beta-gal. The lac-Z gene. Okay. If lac-Z gene is inactive, there will be no blue color. If there is no blue color, see, if there is no blue color formed after the addition of a chromogenic substance, we can assume that there will be no inactivation of lac-Z. Okay, there will be inactivation of lac-Z. If inactivation of lac-Z, we can assume there is an insert. If insert is there, we can assume that is recombinant. Okay, and in this case you can see there is no insert. Since there is no insert, there is no inactivation of the beta lac-Z gene. and it can produce beta-galactosidase enzyme. Since beta-galactosidase enzyme is present, there will be blue color when we add a chromogenic substance. There will be blue color. There will be blue color. Okay, if blue color is there, we can easily predict that this will be non-recombinant. So listen. If no blue color, if there is no blue color formation when we add a chromogenic substance, it is recognized as recombinant. Got it? If there is a blue color, did I say wrong? Come on, once again. If there is no blue color, it is identified as recombinant. If there is blue color, it is identified as non-recombinant. So recombinants are selected in a single step. Okay, so insertional inactivation method is better than antibiotic resistant method to select recombinants. Got it? Okay, listen. Why does the insertional inactivation method to detect recombinant DNA is preferred to antibiotic resistance procedure? Because antibiotic resistant procedure is very easy method. Okay. At the same time, insertion inactivation method is very easy process. Okay, because antibiotic resistant method requires 2 steps. Okay, it requires culturing of bacteria in 2 mediums, one containing ampicillin and second containing tetracycline. Okay, that is a cumbersome process. Got it? Now we can do the previous one. Explain how recombinants and the non-recombinants are differentiated on the basis of color production in presence of a chromogenic substance. Name the procedure. Can you name the procedure? The procedure is selection of recombinants by antibiotic resistance. Selection of recombinants by antibiotic resistance. Got it? Okay, and how it is selected? If there is no blue color, if there is no blue color, it is identified as recombinant. If there is no blue color, it is identified as recombinant. If there is blue color, it is identified as non-recombinant. Got it? Okay. Next question. Non-viral and non-vector methods are sometimes used to transfer gene or alien DNA into the plant cell. Okay. Explain one such method used in genetic engineering. In genetic engineering, there are many methods to transfer foreign DNA into a host cell. Okay, if the host cell is a plant cell, if the host cell is a plant cell, this is a plant cell. Okay, we want to introduce foreign DNA into the plant cell. We can use a method called biolistics or gene gun. Or we can call that gene gun. This is the diagram of gene gun. Okay, see, shooting something into this. What is that something? Shooting microparticles of gold or tungsten. Okay, shooting microparticles of gold or tungsten coated with DNA into the plant cell. Okay, now with the microparticle of gold or tungsten, the coated DNA will also be introduced into the plant cell. This method is called gene gun or biolistics. Got it? Okay. Next one, name the host cell in which micro-injection technique is used to introduce an alien DNA. See, you can see in this figure, this is a cell. To this cell, foreign DNA is injected. to the cell, foreign DNA is injected. This is done in animal cells. Okay, micro injection technique is done in animal cells. Okay, injection of foreign DNA into the nucleus of an animal cell using a micro syringe is called micro injection. Got it? Next one. Which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? See, human lymphocytes. This is human lymphocytes. Human lymphocytes, okay, whatever it is, human lymphocytes or any animal cell. If you want to insert a foreign DNA into an animal cell, you can use a vector. You can use a vector that is retrovirus. Retrovirus, okay. Retrovirus is the vector used to introduce foreign DNA into animal cells. Okay, but if you want to introduce a foreign DNA into a plant cell, you can use another living vector, that is agrobacterium. Agrobacterium tumefaciens. Agrobacterium tumefaciens to use it as a vector to plant cells, especially dicot plants. Okay, but retrovirus is used as a vector to animal cells. Okay, the question will be, name 2 organisms that can be used as a vector, one to plant cell, one to animal cell. Okay, to animal cell we can use retrovirus as a vector, to plant cell we can use agrobacterium tumefaciens as a vector. Got it? One is a virus, one is a bacteria. Okay, next one. What are bioreactors? How are large volumes of cultures maintained and processed in them? Okay, the figure will not be there. The figure I just added for your reference. Okay, see, what are bioreactors? What are bioreactors? I'll write here the definition of bioreactor. Bioreactors. Okay, these are the vessels. These are the vessels, okay, in which raw materials are biologically converted into products. Okay, we are selected the recombinant bacteria. Okay, after that, adding this recombinant bacteria into this bioreactor for large scale culturing. Okay, in this bioreactor the bacteria will be cultured. Okay, after that the bacteria will produce the desired product. Okay, the recombinant protein. Got it? For the growth of bacteria, we have to give many things for the bacteria to grow. First one, they need optimum pH. Okay, they need optimum temperature. Okay, and continuous aeration. Continuous aeration. Okay, constant agitation. Substrate to grow. Vitamins, etc. Okay, the bioreactor provides optimum conditions for the bacteria to grow. Okay, which are the optimum conditions? Optimum pH, optimum temperature, continuous aeration, agitation, substrate substrate and vitamins for the bacteria to grow. Got it? See, in some questions the diagram will be given, the diagram of bioreactor. What is this reactor? This is simple stirred tank bioreactor. Which is this one? Simple stirred tank bioreactor. The labeling question will be there, like ABCD. You have to label those. Got it? So how are large volumes of cultures maintained and processed? By providing this much optimum conditions. Got it? Next one. What is the role of sterile air bubbles in the sparged stirred tank bioreactor? See, this one is simple stirred tank bioreactor. This one is sparged stirred tank bioreactor. In this sparged stirred tank bioreactor, listen, this is the tank. Okay, in this there will be a sparger here. There will be a sparger here. Okay, sparger here. Okay, sparger here. This sparger produces air bubbles. The sparger produces air bubbles. Okay, the sparger produces air bubbles. Okay, what is the advantage of this air bubbles? What is the role of sterile air bubbles in the sparged stirred tank bioreactor? See, this is an air bubble. Air bubble. These bubbles increases the surface area for oxygen transfer. It is given already in this figure. See, bubbles dramatically increase the oxygen transfer area, but this figure will not be given in your question. Okay, question is just this figure I added in addition. Okay, see, this oxygen bubbles dramatically increases the surface area for oxygen transfer. The more the bubbles, the more will be the surface area for oxygen transfer. Okay, next one. Which of the following is not a component of downstream processing? What is downstream processing? After this biosynthetic phase, okay, the processing in the bioreactor is called a biosynthetic phase. We will get the product, we will get the desired product or recombinant protein. Okay, we have to collect that through a sampling portal. After that, the desired product should be processed. Okay, the processes are called downstream processing. First, we have to separate separation of the recombinant protein, purification of the recombinant protein. Okay, adding preservatives to the product. Okay, clinical trials of that product, and quality control tests. These steps together called downstream processing. Downstream processing. Okay, which of the following is not a component of downstream processing? Which one? Separation is there, preservation, adding preservatives is there, purification is there, and expression is not there. So the answer is D. Got it? Now you have finished all the questions. By doing this, we have covered almost all areas of this chapter, biotechnology principles and processes. We will see in another chapter, biotechnology 2. Bye.